Okay, hello everybody. My name is Meenal Datta, and I'm an assistant professor at the University of Notre Dame in the Department of Aerospace and Mechanical Engineering. Today, I'm going to tell you actually about some of our exciting unpublished work in a relatively new area of research in oncology that we refer to as immunomechanics research and mechano-immunology research. And I'll touch on those terms a bit more as we move forward. We're particularly interested to understand these phenomena in the context of very difficult to treat brain cancers, including glioblastoma, which you can see here on this title slide in MRI. So for those in the audience who are unfamiliar with this disease, glioblastoma, which I'll also refer to in abbreviation as GBM, is a universally fatal disease. So patients who are diagnosed with glioblastoma unfortunately succumb within about 2 years of diagnosis. And this disease is resistant to all current forms of therapy and even new therapies, including immunotherapy. And part of the reason that diseases like glioblastoma are resistant to therapy is because of this so-called abnormal tumor microenvironment, which really fuels disease progression and treatment resistance. And so when we think about the tumor microenvironment, it actually contains all of the same components as our normal tissues, but the problem is that these are often abnormal. So for example, there's an overabundance of cells, including cancer cells and the host cells of that particular organ, in this case, the brain, which could include stromal cells of the brain as well as immune cells. Those cells tend to cluster together tightly, proliferate, and then produce this extracellular matrix or protein scaffolding that basically holds all the tumor tissues together. Blood vessels can be present, but they tend to be poorly formed and leaky. In the context of the brain, we don't know much as to whether there's the presence of lymphatics in the actual brain parenchyma. There certainly is in the surrounding skull. But in most other tissues in the rest of the brain, lymphatic vessels are essentially absent from the tumor microenvironment. And so all of these features really coordinate to basically create a hostile environment that promotes hypoxia and immune suppression, and it really promotes the behavior of these tumor supporting host cells, which again can be immune or stromal. Now, these are all essentially biochemical in nature, but the tumor microenvironment also has mechanical or biophysical abnormalities, including properties such as tissue stiffness or tissue microarchitecture that the cells have to navigate through as they grow and spread, but also mechanical forces, including fluid pressure, which is a result of that leaky tumor vasculature, and a force that we refer to as solid stress. And the reason that we call it solid stress is because it is derived and transmitted by the solid components of tumors, so including cells and the extracellular matrix that they put down. And in terms of this mechanical force solid stress, what we know already is that it can promote migratory and invasive behavior in cancer cells. Very importantly, it actually compresses those leaky tumor blood vessels shut so that you lose even more perfusion to the tumor, which really complicates drug delivery and efficacy. And sort of this line of questioning about tumor microenvironment and tumor mechanics has led my lab, the Time Lab at the University of Notre Dame, to start to answer some of these questions about this interface between immunity and mechanics and really difficult to treat cancers like GBM. And so in particular, the work I'm going to show you today is about this question of how does this mechanical force solid stress actually impact immunity and immunotherapy outcomes in glioblastoma. And so when we think about this potential reciprocal relationship between, let's say, mechanical forces and immune cells, we have to consider which immune cells are actually present in the glioblastoma microenvironment. And so I'm showing here an image. This is a cortical cross-section of a mouse brain that's bearing a glioblastoma tumor. You can tell where the tumor is because of the dense area of cells. But actually, all the cells that are in white are myeloid cells. This is the EBA1 stain, so it's picking up both resident microglia, the sort of native immune cells of the brain, but also infiltrating macrophages from circulation. And what we can see if we sort of zoom in, particularly at the interface between glioblastoma in the brain, is that these myeloid cells are highly prevalent. They can make up to 50% of a brain tumor, like glioblastoma, and they tend to exist in mostly what we call pro-tumor phenotypes. This can also be referred to as anti-inflammatory or M2-like or immunosuppressive. All of these are essentially synonymous terms for the same functional behavior. In this case, the red is staining for the marker arginase 1, which is certainly immunosuppressive. And so our questions were really twofold and sort of come back to this question of mechanics and immunity, which was our hypothesis is that because these myeloid cells are present in such large numbers, that they might actually be contributing to that mechanical force solid stress, which again is produced by mostly the cell growth in tumors. So this is really where the immunomechanics part comes in. This is the ability of immune cells to exert a mechanical force. Whereas our second question is the reciprocal relationship. How does that mechanical force actually impact myeloid cell phenotype and function? And so this is where the mechano-immunology comes in, the impact of those mechanical forces on the behavior of those myeloid cells. So we decided, I'm going to show you now all unpublished work, and we decided to focus mostly on macrophages for this for this initial work. You can also see a copy of the work I'm going to show you on bioRxiv if you want to see a preprint. But what we wanted to do in order to really answer those questions mechanistically was create a new 3D myeloid cell model out of these macrophages. So first of all, to the best of our knowledge, it had never been shown before whether these immune cells on their own can actually produce growths, just like they might accumulate in 3D in the body. We wanted to see, can we get them to form a 3D spheroid on their own without any cancer cells present or any supportive matrix? And we found that actually they can. So this is the mirroring cell line raw 264.7. And what we found is that when we grow them encapsulated in an agarose hydrogel, we see that we can get them to form different sizes of spheroids that have also different proportions of live cells, which are shown in green, or dead cells, which are shown in red. But even in particularly stiff gels, here you can see an agarose concentration up to 2%, we still have relatively low fractions of dead cells. So the reason that we picked agarose is because this is a plant-based hydrogel. So these mammalian cells, these macrophages, actually can't break this down. So the only way that they could create a spheroid is basically by growing and expanding and sort of pushing on that surrounding gel. And that's exactly what we have seen in mathematical modeling. So in order to produce these spheroids, the macrophages grow, they proliferate, they push on the gel around them in order to expand. And in softer gels over here on the left-hand side, they're able to expand to larger sizes and to exert higher levels of stress. So this is basically just using first principles of mechanics, including Hooke's law, to estimate these displacement values of the surrounding gel. So the spheroid is modeled here in white in the modeling, and is based off of this confocal immunofluorescent imaging. But as the gel gets stiffer, we see that not only does the spheroid size decrease, but also the extent of that stress that it's exerting is over a smaller area of the gel as well. And moving forward, we can take sort of a broader wide field view of these gels so that we can see sort of the overall phenotype of the spheroids that are formed. And what we see is that these spheroids are dictated in part by these mechanical cues. So as we increase stiffness moving from left to right, we see that the spheroids become much smaller, they become much rounder. Again, it's harder for them to push on the stiffer gel as they grow, but they can still form spheroids in each of these conditions. But what's interesting is if we add some biochemical cues, so for example, if we add LPS, or lipopolysaccharide, which is basically a fragment of a bacteria that these immune cells will respond to, we see that when we quantify spheroids with just increasing gel stiffness, they do decrease, as we see in the immunofluorescent imaging. But when we add this biochemical stimulation to these immune cells, we're actually able to overcome some of those mechanical effects. And so you see, for example, in zero point 0.5 or 1% agarose gels, we definitely get an increase in spheroid size when we add this biochemical stimulation to the immune cells. And we also see something similar happen when we apply a mechanical force. So right now, what I've shown you is evidence that these myeloid cells, these macrophages, can exert this mechanical force solid stress in order to grow and in order to produce these spheroids. But we can also see what's the impact of mechanics on the myeloid cells themselves and their behavior. So for example, if we look just at increasing stiffness, we see that we actually increase the expression of inflammatory cytokines by these macrophages. So with increasing gel stiffness, we see an increase in arginase 1, which is definitely an M2-like marker, or anti-inflammatory or pro-tumor-like. Again, these are synonymous. But we also do see upregulation of INOS, which is M1-like marker, or anti-tumor or pro-inflammatory. So it could be that perhaps there are mixed effects of responses of these immune cells to mechanics, but also we need to understand that it's not so simple just to bin myeloid cells into two separate phenotypes. They likely exist on a spectrum between them, and that might be some of what we're seeing here. So in addition to the surrounding stiffness of the gel, we can also apply a mechanical force to stimulate that solid stress that these macrophages experience within the tumor microenvironment. I'm showing here just a simple graphic of how we actually do this in a transwell system with a cellular monolayer, but we can do the same thing with spheroids as well. Again, you can refer to the preprint if you want to see exactly how we did this. But with increasing stress, we see really similar phenotypes. If we compress the cells and exert the solid stress on the macrophage spheroids, we again see a pretty profound increase in that M2-like anti-inflammatory behavior, but also a little bit of an increase in that M1-like pro-inflammatory behavior. And so this is all very interesting and mechanistic, but of course, eventually we want to be able to use this type of information to inform treatment strategies. Can we actually target some of these interactions between the immune system and the mechanics of the tumor in order to improve treatment outcomes in cancers like glioblastoma? And what I'm showing here is sort of that typical professor behavior where you summarize an entire paper on one slide. But this is published. The reference is down here at the bottom of the slide if you'd like to refer to it for further information. But I just wanted to highlight some key points that relate to this question. So we actually found that using a very simple, safe, and affordable antihypertensive treatment called losartan can actually achieve this effect. So this is a drug that is FDA approved, and we repurposed it not for its blood pressure control properties, but actually it has anti-matrix properties because downstream of the molecular target of this drug is actually matrix controlling molecules like TGF-beta. And connective tissue growth factor. And so without going through all the details, I'll just briefly say that when we apply this drug, losartan, to mice that are bearing these orthotopic glioblastoma tumors in the brain, we see that we can actually reduce the amount of matrix, for example, the matrix molecule hyaluronan, also known as hyaluronic acid. We can improve perfusion. I'm showing you an example of live angiography through a cranial window in mice. So you can see under losartan treatment, we tend to have better perfusion of those red perfused blood vessels. They also look much more normal and straighter compared to the control tumors. When we run flow cytometry, we see that we actually reprogram the entire myeloid compartment. Again, for the sake of time, I'll keep this brief and say we mostly reduce those M2-like or anti-inflammatory pro-tumor-like microglia, which is abbreviated as MG, macrophages, which is abbreviated as TAMs, and myeloid-derived suppressor cells, which is abbreviated as MDSCs. And finally, we see an enhancement with this drug in the cytotoxic T cell ratio to regulatory T cells, which we also confirmed further with single cell sequencing. So if you refer to the paper, you'll see that the next thing we did is we actually combined this drug losartan with immune checkpoint blockade in mice for survival studies and found that it was actually able to cure about 20% of mice that were bearing these glioblastoma tumors. And we think that a large part of this mechanism is overcoming these immune mechanical features. So this is very exciting. The last bit of unpublished work I want to touch on today is more taking a step back and thinking about the way that we model these interactions to better study immunomechanics and mechano-immunology and glioblastoma. And this is really a new area of development for our group. And so when we think about this, we have basically, oh, excuse me, I'll move back one. We have basically three options that are common, especially in academic labs for studying, let's say, these interactions between cancer cells and myeloid cells like macrophages and glioblastoma. So the most common are to use 2D monolayer cell culture, and these are cheap and reproducible. But it can be really difficult to study more than one cell type. And of course, studying cells in 2D doesn't always recapitulate the biology of the human. We can also use animal models. For example, the previous study I showed with losartan certainly used the mouse model of glioblastoma. These are great because, especially for immunology, they do allow for the full immune competent system to be considered in studies of immuno-oncology. But there are some limitations, including cost. They don't always recapitulate the human biology, depending on what animal or disease you're studying. And of course, the most recent Modernization Act from the FDA has basically called on the community to start to reduce or replace the use of some of these models before we move into clinical trials with patients, which is certainly not trivial. But one of the ways in which researchers are trying to overcome these limitations is to move into these 3D organoid cultures. And really the only difference between an organoid and the spheroids that I showed you before is that an organoid is going to be comprised of more than one cell type. So for example, combining a cancer cell with a myeloid cell. And so some of these advantages are that you can model many different organ systems, you can model different types of diseases, including cancer, but others. But since it's still an ex vivo system, you can also do mechanistic studies, including genetic manipulation. Because they're small and reproducible, they're great for high throughput drug screening as well. So in our lab, we actually use multiple methods to try to culture these organoids. For the sake of time, I won't describe them here, but each of these, none of these approaches is new. They're all established in the literature, and each has their own sort of advantages and disadvantages. But one of the big disadvantages, no matter what type of culture system we're using, is that on Earth, when we create these organoids, they actually suffer from a lot of effects due to Earth's gravity. So we see such impacts as settling or sedimentation of these spheroids or the cells to, let's say, the bottom of a cell culture dish. We can see them start to disaggregate. And as you can see in this image that I'm showing here, the biggest problem is heterogeneity. We see non-uniformity in the size of these spheroids, in the shape. You can see in this image some are perfectly round, others are sort of football shaped. The viability, in this case, we're showing images of live cells in green versus dead cells in red. And also that cell incorporation. If we're really trying to get multiple cell types, this can be a problem with organoids grown on Earth. And that's led us into a very sort of literally out there approach, which is to try to use microgravity environments to better support this robust organoid growth, specifically on the International Space Station. And so this is not a new idea. This has been around now for a few years with other types of researchers trying to grow organs and organoids in microgravity. You can see a depiction here of an astronaut on the International Space Station growing organoids for Cedars-Sinai. And there are multiple advantages here of growing these organoids in microgravity. First of all, the self-assembly of these organoids is rapid. It doesn't require external scaffolding like that agarose gel that I showed you on previous slides. You get much more uniform organoids. in size, shape, viability, the incorporation of those cells. We really need them to be uniform if they're going to replace, let's say, a mouse model that are genetic clones of each other. You can have much more complex cell-cell interactions and better robustness and reproducibility. And so this is my last research slide here. I just want to show we currently actually have an experiment on the International Space Station as we speak. So it's actually going to come back to Earth in a couple of days now. So this is very exciting for us. What we're trying to do, I apologize that the image is not showing up here on this slide, but you can imagine that the organoid actually looks exactly like the one shown here on this previous slide. So you could imagine instead that the green are the cancer cells and the orange are myeloid cells. And that's exactly what our spheroids look like as well in this missing image. But really what our goal is to synthesize these cancer myeloid organoids in microgravity on the International Space Station and to compare sort of the growth and behavior of those organoids to ground-based models here on Earth using a variety of methods, including RNA sequencing, staining, and proteomics as well. We partnered with a company called Space Tango, which uses multiple partners for launching, including SpaceX. And as I mentioned, this has been on the International Space Station for about a month now. This uses Space Tango's patent pending technology called the QubeLab, which allows us to grow these organoids. And in the future, what we're hoping to do is once we can actually characterize these organoids, we plan to further develop them for drug testing for these myeloid reprogramming agents, but also maybe drugs that target these interactions between mechanics and immunity as well. So that's actually the end of my talk. I'd like to thank the audience for listening. I'd like to thank all of our funding support, my students from the Time Lab who actually did the work. And if anybody has any questions, you're welcome to email me. My email is provided here, and you can learn more about the work that we're doing on our website as well. So thank you for your time.